Hi, my name is Neena Haider, and I am an associate professor at Harvard Medical School, and my lab is housed at Schepens Eye Research Institute, which is part of Mass Eye and Ear. So my expertise is genetics. I'm a geneticist. So I was trained in a human genetics lab where we identified probably 15 to 20 different mutations associated with human diseases. And I was part of the Human Genome Project, and so sequenced chromosome 15q22. And my research focus is a bit broad in a way. I study the retina, which is the back of your eye, it's like the camera of your eye. And I study all retinal diseases. I did my postdoctoral training at the Jackson Laboratory because I wanted to expand the research in what we could do with human genetics. So there we learn what's the disease, and we can see it in the patient, but then to really come up with a therapy, which is my goal, we really have to develop a model system. So I have probably 6, 7 different models for AMD. And the way I approach AMD, or actually all of my studies, is kind of three-pronged. One is we want to know what causes the disease. So that's at the DNA level. We want to understand disease pathogenesis and progression. So what's wrong in the retina? When does it go wrong, and how much does it progress? How fast it progresses? And you need all that basic discovery science information to really develop a viable therapeutic. We have actually recently discovered the only, that I know of, multigenic genetic model for AMD. Why is that so exciting? Because AMD, thus far, we have no idea what causes the disease. We know aging is a huge factor. We know probably 50 or some odd different genes that are associated. They're risk factors, but they don't tell you this will really cause it. So this is exciting because we know there are multiple genes as well as the environment that play a role in AMD, but no one's ever said this disease is caused by XYZ genes for sure. You have those mutations, you'll get. So we have one of the first models where it requires these multiple genes going wrong, and then you get disease. So that discovery will be huge and game-changing in terms of developing a viable, effective therapy. So our goal for AMD is we want to prevent disease. The best is prevention, right? Prevention is the best medicine. And we're doing a few different things for that. One is really exciting, is our retina in a dish. So we're just starting this project where we're going to take blood from patients and grow a retina from their own blood. So it's your retina in a dish. And there's so many things we're going to be able to do with that. We're going to be able to test our novel therapies we develop as well as other current therapies that are out there. It brings precision personalized medicine to the table where we can say, for you, this is the best therapy, right? And expanded from that, which is even more exciting, but down the pipeline, think maybe 5, 6 years down. Everything used to be 10 years down. Now it's like 1, 2 years. It's game-changing. The research is exponential. What we want to do is make the retina, and when we grow them, this is like in a year or so, we'll have something maybe when you were in your teens. So that's a healthy retina. If we can get that made from you, if we can put that back in as a therapy, then your eyes could, the goal is, then that would be as healthy as when you were back in your 20s, before you had disease ever. We're doing the retinas in a dish now. So this is a lot of my work that we do is discovery science. And discovery science leads to translational, transformative medicine that leads to the clinic. How soon? There's just so much research going on into making these organs in a dish that I don't think it'll be that many years before we can go into the clinical trials. I actually have a US patent issued, not approved, but actually issued this year on our modifier gene therapy, and that includes these class of genes. They're called nuclear hormone receptors. What they do is regulate everything. So I like to say, like, we should do externally, but internally, every cell in your body is trying to do the same thing. It stays zen. It's trying to find its homeostasis or zen state, right? And what we have patented is we have the genes that help you do that. And one of them is really associated with AMD. And we have a couple others that regulate that. So we're able to regulate these hundreds and hundreds of networks of genes that all come together to stabilize the situation. And our goal for that with clinical trials for AMD would be in less than 5 years, that we could test that therapeutic to say, can we, before you get disease, can we stabilize the situation as soon as we find out there's something going on? And that and those people that have, family history, risk factors, that you can see down the pipeline, I might have this happening. You get more frequent tests done, and we work very closely with the clinicians. And then the idea would be say, okay, we'll timeline you and start a therapeutic before you start seeing a problem. Because really, what the clinicians are seeing as a problem, the drusen, the spots, and later on the blood vessels that leak, and that's what causes your vision problem. That's like something that's easy for you to see. And I give the analogy of, if I look up in the sky on a nice bright day, not like today, you can see certain stars, but you can't see the whole galaxy, right? You can't see all the stars even within our own galaxy. So just like that for AMD, while we see a few of the key features, there's many others on a DNA molecular level that are happening that we're not visualizing with our naked eye and our instruments externally. But in the retina dish, we can visualize those. And by visualizing those early on, we can start to be able to track and say, okay, this person is down that path that we need to think about a treatment for. So we work very closely with Dr. McDeangelis, who I know you know, and weekly we converse about these networks and networks of genes and how they interplay with each other. So if she finds something in the patients, in the donor eyes, in the patient database, then we can model that. And we can say, okay, because when you make a model, then you can test the questions you can't test in humans, which is, if I add this drug, what happens? If I add this therapy, what happens? If I take away this, if I manipulate the system, how is it going to impact? And those are the type of studies that we do then.